Hi there, my name is Mike Pishvaian. I'm an associate professor at Johns Hopkins University, and it's my pleasure today to talk about the relevant updates in GI cancer from ASCO 2023, incorporating some of the data that we've also seen throughout the year. I'm going to talk a little bit about some of the targeted therapies and some of the more practical aspects of caring for patients with GI cancers that have come up in the last roughly 12 months. There was a lot of excitement focused around targeting KRAS in pancreatic cancer. And this excitement is worthwhile because most pancreatic cancers harbor KRAS mutations. And really, the KRAS mutation is a key event in the initiation and the development and the growth and the progression of pancreatic cancers. So just to review what happens with the KRAS mutation, KRAS actually, the enzyme cycles in off and on position, and the on position actually leads to downstream signaling that can lead to many mitogenic and pro-growth signals. In this context of a KRAS mutation, the KRAS mutation can lead to this being a constant on signal where there is constant signaling for the cancer cell or the cell to grow and divide and spread, et cetera, et cetera. So it's been a key target to try and shut down KRAS, shut down the KRAS enzyme for pancreatic cancers in the hopes of actually slowing cancer cell development and growth and everything else. The problem is that KRAS has been very difficult to target, and there's been over the years attempts to target KRAS, and we're starting to see some headway being made in this way. So, for example, the KRYSTAL-1 study, which was originally presented by Dr. Saab at ASCO GI about a year ago, and then Dr. Pont presented an update at ASCO 2023. The KRYSTAL-1 study is a trial that looked at adagrasib, a KRAS G12C specific therapy in patients with pancreatic cancer. So KRAS mutations are common in all pancreatic cancers, but in pancreatic cancer, only about 2% harbor the KRAS G12C mutation specifically. And just to be clear, this drug only works for KRAS G12C. It does not work for other mutational subtypes of KRAS. So the way adagrasib works is it covalently binds to KRAS G12C in its GDP off state, and thus locks the KRAS for that cell in its off state, preventing further KRAS activity. And the phase 2 monotherapy trial in solid tumors looked very promising. It was mostly pancreatic cancer patients, 21, as well as other GI cancer patients, and all patients had to have had at least one line of prior therapy. And you can see by the waterfall plot and by the response rate that this actually did show efficacy. Adagrasib showed an objective response rate of 35% overall across all disease types, all GI disease types, as well as specifically 33% in pancreatic cancers and 42% in biliary cancers. The waterfall plot suggests that there were also several other patients who had some degree of tumor shrinkage, although it did not meet the criteria for RECIST criteria response. And in fact, when you look at the waterfall plots specifically for pancreatic and biliary cancers, you see again this 33%, this 35% response rate in the patients, and then disease control rate was upwards of 86%. Adagrasib does come with some side effects. There were no grade 4, grade 5 adverse events, but nausea, vomiting, diarrhea, sort of GI-related toxicities were seen in about 40 to 50% of the patients. Fortunately, most of them, or almost all of them, were grade 1 or grade 2. There was also a grade 3 fatigue that was seen. So this is something that's going to have to be explored, but this really may open the door to targeting a specific mutational subtype of KRAS in GI cancers across the board, and specifically in pancreatic cancer. Now, I think some of the greatest promise in targeting KRAS is going to be in combination therapies. We know that the KRAS inhibition can be effective in some patients, but actually, Rona Yeager showed in a publication in spring of 2023 that the combination of adagrasib and cetuximab, an anti-EGFR therapy, in KRAS G12C mutated colorectal cancer significantly improved objective response rate. This is from her paper you see on the right-hand side there. And the waterfall plot suggests that adagrasib alone definitely did lead to some improvement. She didn't bother to test cetuximab alone, which makes sense because we know the response rate in a KRAS mutated colorectal cancer is 0%. Adagrasib alone led to a response rate of about 23%, but the adagrasib plus cetuximab combination became the response rate was 46% with a 3-month improvement in median overall survival. And this is really because what you're doing is you're creating a cell that is now functionally KRAS wild type and using an anti-EGFR inhibitor. So that's really where the promise comes. Could other combinations also be used to try and target both shutting down KRAS and then also targeting other downstream or upstream signaling molecules? Along those lines, there was also another abstract that was presented at ASCO. This was very early in the abstract. It was just a phase 1 study, but it was with a SHP2 inhibitor. And the SHP2 inhibitor was also tried in several cohorts with other combination therapies. And true to this idea that you actually need combination therapy to really achieve a significant response, one of the only patients that actually responded in this trial was the patient that had a combination of both the SHP2 inhibitor and encorafenib and cetuximab in a BRAF V600 mutated colorectal cancer patient. So I think this was just one patient. I don't want to make too much of it, but the point is that combinations may be the way we need to think about exploring KRAS inhibitors in the future. And then finally, you can't talk about ASCO 2023 without really focusing on the KRAS personalized vaccines. This was presented by Dr. O'Reilly, as you see here in the bottom. This drug, this vaccine was all over the news because of the promise that it's been showing, and I think justifiably so, although right now, the only data we really have is correlative and translational. So this trial was a KRAS G12D and G12R, as well as TLR9 adjuvant targeting cancer vaccine. It was given for patients who were very specifically defined, those who had no evidence of cancer visibly, but had some suggestion of cancer still being present, either because they had minimal residual disease on a ctDNA assay or the serum biomarker such as CA19-9 was elevated. So patients who fit into that category were then given a prime and immunization of this vaccine over several vaccine doses over a period of time. They were followed with serum biomarkers as well as ctDNA and tumor biomarker analysis. And what they showed, again, just as a translational endpoint, was that there was a 77% reduction in the biomarker/ctDNA profile, and focusing specifically on ctDNA, there was actually a 32% clearance of ctDNA for these patients that received this vaccine. We certainly need to wait to see how this data matures, particularly in the clinical outcomes, the relapse-free survival for these patients. And there is an ongoing phase 2 trial that's about to be launched for this patient population, again with a hard clinical endpoint that'll give us a much clearer picture of the clinical efficacy for this agent.